Hello, I'm Steve Potter. I'm an associate professor in the Department of Biomedical Engineering at Georgia Tech and Emory University School of Medicine. My lab is one of 9 labs that are part of the Laboratory for Neuroengineering at Georgia Tech, also called the Neuro Lab. And I'm also a member of the IBB. Our research is aimed at the basics of learning and memory and how information is processed in the brain. I brought my model brain here. Let's see here. So the kinds of questions that we're asking are, what changes about your brain when you learn something? What physically is a memory? How is it that you remember and store memories? How do we forget memories? Surprisingly little is known about these processes. In the year 2000, we created a new approach to studying these questions called embodied cultured networks. We grow cultures of a few thousand neurons and glial cells on multi-electrode arrays like this. So these are the kinds of cultures. You can see here brain cells wiring up. Those include neurons and glia. This movie takes place over the course of about 1 week in culture. And there's the culture dish that I was holding up. These dishes have about 60 electrodes embedded in the substrate, and the neurons that are lying near the electrodes can be recorded, and we can also electrically stimulate them. So we can form a two-way connection between a computer and the neurons there using these multi-electrode array culture dishes. So we created the hardware and the software that we need to make the first closed-loop system between a multi-electrode array culture and a computer. Here's an overview of the lab. We have a culture dish sitting here in electronics inside of a microscope, and the signals that are recorded by this electronics are sent through these wires to the computer, and then the computer can send stimulation commands back to the culture. So that's the closed loop. You could think of the recordings as being motor control information and the stimulation as being sensory information. So we can feed information into the cultures, and we can record information out of the cultures. We can also use that information that we recorded to control a simulated animal on the computer called an Animat, or we can use it to control a robot. And we call those robots Hybrots because they're hybrids of living neurons and robotic artifacts in one semi-living system. So for example, in collaboration with Symbiotica in Perth, Australia, that's why we have this Australia thing on our map here, there's Perth, they built this robotic arm that draws, and we hooked it up to neurons in our lab. So there it is drawing, and this robot has traveled all over the world. My lucky former graduate student Doug Bockham got to go to many different exotic locales where it was exhibited and controlled all the while by neurons in our lab talking to it over the internet. There was also a video camera that was filming the drawing process while it happens, and that information would get sent back to our lab and was translated into electrical stimulation patterns that were delivered to the network. So again, we have this closed loop system in which we're recording signals, we're controlling something with it. It has a sensory system, and that gets translated back into stimulation that's delivered to the culture. Okay. So there's the closed loop system again. Cultured networks have advantages for studying learning and memory compared to intact animals. Probably the most important advantage is that they're much less overwhelming in their complexity than the human brain or even than a rat brain or a mouse brain. This has about 10 trillion neurons. Our cultures have about 10,000 neurons. So a few orders of magnitude there. Simpler. most of the circuitry that makes brains do what they do is missing from our cultures, but we believe that a good fraction of the interesting dynamics of neural networks are also present in the networks that we grow in these culture dishes. So the other big advantage that cultured networks have is that they're very accessible to optical microscopy and other interventions that you might want to do, pouring on pharmacological agents or electrically stimulating. That sort of thing is much easier to do in vitro than it is to do in vivo. So for example, we built a 2-photon microscope in the lab, and we can use it to make these time-lapse movies of neurons forming connections. So if you look carefully, you might be able to see some growth cones reaching out and forming brand new synaptic links between two neurons. So we're hoping to use this technology, such as 2-photon microscopy, to watch the learning process while it happens. Cultured networks, if you record their activity, they produce barrages of activity called population bursts. Here's an example of one of those. So that is a population burst consisting of thousands of action potentials that the neurons have fired in the course of about half a second or so. And this movie is slowed down by a factor of 10. These bursts resemble epileptic seizures, and we developed techniques for stimulating neural circuits in culture using the multi-electrode arrays that they're growing on. So we distribute sprinkling of electrical stimulation across the network, and that quiets down these bursts of activity. So since we were successful in quieting seizure-like activity in our culture dishes, we've also begun a collaboration with Bob Gross, who is a neurosurgeon at Emory University School of Medicine, to test out this idea as a potential drug-free treatment for epilepsy. So we're hard at work first testing it out in animal models, and if that's successful, we'll then move on to testing it out in patients that have intractable epilepsy. And that project was funded by the Coulter Foundation. So our closed-loop systems allowed us also to develop techniques to train cultured networks with electrical stimulation. And this slide is an example of that. Let's see if I can get it to go again. So here we have a neurally controlled Animat running around in a field. And when the movie started, it had already learned to stay in the middle of the field. And then we confused it by reversing the orientation of its sensory system. And you can see that after a few minutes, it learns again how to move back to the center of the field and to stay there. So this is all thanks to multi-electrode electrical stimulation being delivered to a real live cultured network in vitro. So this technology has also allowed us to uncover some previously unknown learning mechanisms, such as shifts in the dynamic attractors expressed by these networks. These bursts and other activity patterns can be looked at as attractors in which the activity tends to flow down certain circuits. And we were able to stimulate these cultures and cause the activity to flow down different circuits. It's thought that synapses which are the connections between neurons, that they play an important role in learning and memory. And the learning process is thought to be in many ways thanks to changes in the strength of those synapses. We discovered that changes in the propagation of these neural signals down axons, which are the tiny fibers that make up nerves, such as in the brainstem there. We discovered that propagation of neural signals down these tiny fibers, called axons, is also involved in neuroplasticity. Propagation plasticity in axons might be an important non-synaptic learning mechanism. So this was made much easier to study by the simplicity of our cultured networks and by the fact that we have 60 electrodes through which we can both stimulate and record them. So our lab is an interdisciplinary lab, encompassing not just neurobiology, but also a lot of engineering, electrical engineering, mechanical engineering, software engineering. We do computer modeling, we do robotics, we do optical microscopy, probably a few other things. So I would say that we're one of the more interdisciplinary labs in the department in the spirit of the IBB. You can learn a lot more about the science and the technology that we are working on by just Googling Steve Potter. All of our papers can be downloaded by going to our publications page. And if you have questions, of course, feel free to send me an email. Thank you.